In this module, we will study what is the experimental evidence available that the transposon, they contain inverted terminal repeats. Remember, this is a very classical statement that the transposon, they contain inverted terminal repeats and has a great significance in the molecular biology. Now, the Stanley Cohen, which was later awarded the Nobel Prize for this discovery, he provided one graphic demonstration of inverted repeats at the end of the transposon with the experiment which are shown. He carried out the experiment. In this slide, the Stanley Cohen, actually he provided a demonstration and a graphical demonstration that how the transposon, they have got the repeated sequences. He took, there is one transposable element, that is a plasmid, which is inside in this circle. Then there is another one, red one, outside with the inverted yellow plasmids there. They both replicate and divide. And when they replicate and divide, there is an intrastrand annealing. जैसे वो अलग होते हैं, single strand में बन जाते हैं, तो there is a annealing occurs and we get a loop over there. The two are formed. And he started with a plasmid containing a transposon with the structure shown here, as we have seen in the previous slides. Now, the original plasmid that was linked to the ends of the transposon. ये transposon के साथ उनको जोड़ दिया गया, which were inverted repeats. ये inverted repeats discover हुए, ये हुए inverted repeats. And what happened that after this finding, Dr. Cohen, he reasoned that if the transposon really had inverted repeats, at its end, he could separate the two strands of the recombinant plasmid. अगर वो वाकई inverted repeats हैं, तो ये दोनों जो हैं, ये separate हो सकते हैं repeats of the plasmid. And what he did, that he get the inverted repeats on one strand to base pair, which either forming a stem loop structure. एक stem loop structure जो है, उसमें उसे वो repeat जो हैं, वो मिल गए. The stem would be double-stranded DNA composed of the two inverted repeats, and the loop would be the rest of the DNA in the single-stranded form. जो loop है, वो एक single-stranded form में है, whereas the other, the stalk, it is a double-stranded form, double-stranded DNA. Typical insertion sequences have somewhat larger inverted repeats from 15 to 25 base pair. यानी कि typical insertion जब होती है, तो उसमें जो repeats हैं, they are 15 to 25 base pair long. Larger transposon can have inverted repeats which consist of hundreds of the base pair long, जिसमें के सैकड़ों बेस पेयर लॉन्ग हो सकते हैं जो कि उसका स्टॉक बनाते हैं.